Hello, welcome to the Chocolates class. In this class, today we are going to discuss about muscular tissue and nervous tissue. How many muscles do we have? There are more than 650 named skeletal muscles are present in our body. According to some sources, their number may reach 840 muscles. On average, skeletal muscle number is 639. Approximately, there are 639 muscles are present, which accounts 40 to 50% of our body weight. Smooth muscle number is countless, and they are distributed in various organ systems. Let us understand the characters of the muscular tissue. You know that study of muscles is called myology, and these muscles are formed from mesoderm. For this there are some exceptions. Iris muscles, ciliary muscles, and erector pili muscles are ectodermal. Except these, rest of the muscles are mesodermal in origin. The other properties of muscles are excitability. When they receive the stimulus, they can respond to that stimulus and get excited. That is called excitability. And they carry that impulse to the various target regions. So that is called conductivity. The main property of the muscle is contraction and relaxation. In addition to this, the muscles show extensibility and elasticity. Look here. Here, when the muscle is contracting and relaxed, when weight is carried by the hand, the muscle is stretched, so it has extensibility. One more property is muscles do not divide after their formation. For this also, exception is there. Some uterine smooth muscles show cell division. Before understanding the various types of muscles, we have to know the jargon or the terminology of the muscles. A muscle cell is called a sarcocyte. Here we have shown one sarcocyte. And the plasma lemma is called sarcolemma. The cytoplasm is called sarcoplasm. And mitochondria are called sarcosomes. Endoplasmic reticulum is known as sarcoplasmic reticulum. The muscles are three types: skeletal muscles, smooth muscles, and cardiac muscles. This classification is based on the occurrence of the muscle or the distribution of the muscles in the body. Some muscles are associated with the bones or the skeleton. These help in the movement and locomotion. These are called skeletal muscles or striated muscles. The smooth muscles, these are present in the visceral organs. These are helpful for the movements inside the organs. Next, cardiac muscles. These are present in the heart. Now, let us see what is a muscle. So, look this one. This muscle is a skeletal muscle attached to the bone by one connective tissue structure called tendon. It is formed by dense fibrous connective tissue, and this muscle is covered by one connective tissue layer that is called epimysium. And this epimysium surrounds some bundles called fascicles. This is a fascicle. A fascicle is a bundle of muscle fibers. A fascicle is covered by perimysium. This is the perimysium. And inside the fascicle, many muscle fibers are arranged. Around the muscle fiber, a thin layer is there, connective tissue layer, that is called endomysium. And after that, we can see the true muscle fiber which is a sarcocyte or a single cell. Here, the muscle is covered by outer layer called epimysium. And one term is there, one term is used in our book, fascia. What is the difference between fascia and the epimysium? Let us see. Epimysium is a dense fibrous tissue that encloses entire muscle. And you can see this. This is the epimysium which is covering the entire muscle. Okay, this is also, you can see this epimysium in this part very clearly. And fascia is a band of connective tissue beneath the skin that separates the muscles from internal organs. That is the major difference between the epimysium and fascia. Epimysium forms a protective sheath around the muscle, and fascia transmit mechanical tension produced by the muscles. Epimysium is continuous with other connective tissue, but fascia has a loosely packed sheet of collagen. These fascia are three types based on their position: superficial fascia, deep fascia, and visceral fascia. Superficial fascia lies beneath the skin. Deep fascia surrounds the individual muscles. Visceral fascia envelops organs and tissues. Now, let us try to understand a striped muscle. These are the several striped muscle fibers. As we discussed earlier, a muscle fiber is a sarcocyte, that is, one muscle cell. These muscles are cylindrical and these are unbranched. These have many nuclei. Multinucleated condition is seen. That condition is called syncytial condition. What is syncytium? Multinucleated condition that is formed by the disappearance of cell membranes is called syncytium. Let us see this one. Syncytium is a multinucleated condition that is formed by the disappearance of cell partition. These are the cells which are arranged side by side. Each cell has a nucleus like this. In the development, cell membranes between two cells disappear. Thus, a multinucleated condition is formed. So, a multinucleated condition that formed by the disappearance of cell membranes is called syncytium. All striped muscles are syncytial muscles. They have dark and light bands arranged alternatively. Look this one. This is a dark band followed by a light band, dark band, light band. In this way, the dark and light bands are arranged alternatively. Dark band is called A band. This is the A band. The light band is called I band. A means anisotropic band. I means isotropic band. These muscles, they do quick contractions and relaxations. These undergo fatigue. Fatigue means after repeated contractions, the muscles fail to contract temporarily, and that condition, that temporary state, is called fatigue. In other words, we can say that the muscles get tired. That is called fatigue. These have very limited regeneration. As we discussed earlier, the muscle cells do not divide, but these can regenerate. This is because of satellite cells, myoblast cells associated with them. In these muscles, myoglobin is present in red fibers, and in the white fibers also myoglobin is present, but very little, and the red fibers have more myoglobin. And these striped muscles are also called skeletal muscles, voluntary muscles, are striated muscles. So all striped muscles are voluntary muscles. Next one, smooth muscles. In our body, their number is countless. These are present within the visceral organs, and so these are also called visceral muscles. These are not under the control of the organism. Automatically, these are maintained by the autonomous nervous system. Such type of muscles are called involuntary muscles. Involuntary muscles means smooth muscles. It is an synonym for the smooth muscle. These are fusiform or spindle shape. You see this muscle cell. Fusiform or spindle shape means both the ends are tapering, the middle part is broad. Such a shape is called spindle shape or fusiform shape. It has a single nucleus. In these muscles, A band, I band, dark band and light bands are absent. That's why these are called non-striated muscles or smooth muscles or unstriped muscles. In these, the contractions occur very slow and they are prolonged. That's why these do not undergo fatigue. In these, myoglobin is absent. These are the only muscles without myoglobin. Cardiac muscle has myoglobin, and striped muscles or skeletal muscles also have the myoglobin. And the cells are connected by the gap junctions. In the epithelium, we discussed about the intercellular connections. Assume that this is one cell, this is another cell, or another muscle cell. These two are joined by one gap junction. Through these gap junctions, ions can pass from one cell to another cell. So these muscle cells are connected by the gap junctions, so ions can pass. So when a muscle contracts, one muscle cell contracts, that will spread to the next muscle cell. The synonyms for the smooth muscles are visceral muscles, involuntary muscles. Examples for smooth muscles are ciliary muscles, erector pili muscles of hair follicles. When the erector pili muscles contracts, goosebumps will form or the hair will erect. These ciliary muscles, erector pili muscles, these are ectodermal in origin. The third variety is cardiac muscle. If you see this cardiac muscle, these are also cylindrical muscles, but these muscles are branched. So branched cylindrical muscles are cardiac muscles. These are somewhat short muscles when we compare with the striped muscles. Striped muscles or skeletal muscles also cylindrical, but those are unbranched, but the cardiac muscles are branched. These have a single nucleus, mononucleate. See, this is one cell, this is another cell. Each cell has one nucleus. And in intermediate board book, it is mentioned that they may be binucleate. Means two nuclei may present. One or two nuclei is given, but in the NCERT book, it is mentioned that these are single nucleated cells. These also have dark and light bands like striped muscles, but these dark and light bands are not distinct. So indistinct dark and light bands are present. Their contractions are rhythmic. So cardiac muscles are also called rhythmic muscles. And because of rhythmic contractions, these do not undergo fatigue. This is the similarity between the cardiac muscle and smooth muscle. Like skeletal muscle, these also have myoglobin. But in this one, myoglobin is more, and gap junctions are present. This character we found in the smooth muscle also. Like a smooth muscle, these are involuntary in function. So when we see the similarity with the smooth muscle, involuntary and gap junctions are present and a single nucleus. And when we compare with the skeletal muscle, these are cylindrical, dark and light bands are present, myoglobin is present. And what are the unique characters of these? The branching is one of the unique character, and next one, rhythmic contractions also unique character. These have intercalated discs. What are intercalated discs? These are the thickened areas of sarcolemma, that is the plasma membrane of two cells. They merge and form a thick structure called intercalated disc. These intercalated discs, they bear gap junctions. Through these gap junctions, ions can be exchanged between two cells. So, these cardiac muscles acts as a functional syncytium. Functional syncytium means even though they are different cells, the cells are isolated with one another, but these contract or relax at a time because of exchange of ions between two sarcocytes. So, cardiac muscles act as a functional syncytium. And one more important character of these muscles is these are auto-excitable. They no need to get a stimulus from outside for its contraction. These muscles automatically they generate the contraction and relaxations. For example, let us take the heart. This is the heart. You know that the heart has four chambers, two atria and two ventricles. In the right upper corner of the right atrium, there is a nodal tissue called SA node is present. It is auto-excitable structure. It generates the contractions and relaxations which pass to the one more nodal tissue called AV node. From here, this impulse is conducted by bundle of His to the Purkinje fibers, and these Purkinje fibers are distributed throughout the ventricle. Thus, the conducting system of the heart is auto-excitable. What the conducting system includes SA node, AV node, bundle of His, and the Purkinje fibers. So now, what are the unique characters of these muscles? Once again, we'll check. Presence of intercalated discs and the presence of indistinct dark and light bands. And these are the branched muscles. These are the special characters. These do not undergo fatigue. Rhythmic contractions they do. And these are involuntary muscles. These are some special characters of the cardiac muscle. Next, nervous tissue or neural tissue. The nervous tissue is formed from ectoderm, derived from ectoderm. Intercellular substance is absent. Like connective tissue, there is no intercellular substance or the matrix. The neurons conduct the impulses. These cells do not divide like skeletal muscles or the other muscles. Some special cells are there, neuroglial cells, which are non-conducting cells. They act like a packing tissue. They undergo cell division. So neuroglial cells can divide, but not the neurons. And in the neuroglia, there are a kind of cells are there, phagocytic cells, microglia. These microglia are mesodermal in origin. and the rest of the neuroglial cells and neurons are derived from ectoderm. Now, the classification of nervous tissue. This tissue is divided into two types, neurons and neuroglial cells. Neurons are the longest cells in the body, and these neurons are several types. Those are multipolar neurons, bipolar neurons, unipolar neurons, pseudo-unipolar neurons. As we discussed earlier, the neurons do not divide. These conduct the impulses from one part to one region to another region. That is the role of the neurons. Neuroglial cells, these are the packing cells, and these neuroglial cells are they can undergo cell division, and these are also ectodermal in origin except the microglial cells. Those some neuroglial cells are Schwann cells, which are present around the axon of the neuron. Oligodendroglia, asterocytes, ependymal cells, microglial cells, these are present in the CNS, and this is present in the PNS, peripheral nervous system. This is present inside the central nervous system. Now, let us take a neuron. A neuron has two parts. One is the cell body, or it is also called cyton, and protoplasmic projection. First, cyton or cell body. And in this cell, cytoplasm is called neuroplasm. It has a single nucleus. It contains cells bodies and neurofibrils. In addition to this, they have lipofuscin granules also inside. The Nissel's granules or Nissel's bodies are the aggregations of the RER, rough endoplasmic reticulum, that is. From this cyton is also called soma. From this protoplasmic projections arise. Some are short, branched, numerous. These are called dendrites. These dendrites also have the cells bodies and neurofibrils. These dendrites bring the impulse from outside to the cell body. So, dendrites are efferent fibers. Next one, axon. Axon is the longest protoplasmic process of the nerve cell. This axon may have some side branches. These are called collateral branches. We should not get confused with the term collateral glands. Collateral glands are present in cockroach, and collateral filament or branch is related to the neuron. The neuron's axon side branch is a collateral branch. And these axons, these are with the neurofibrils, but Nissel's granules are absent in the axon. The terminal part of the axon has many branches. These are called telodendria or terminal branches. Each branch ends with a button called terminal button or presynaptic knob. These presynaptic knobs are rich with mitochondria. Associated with this axon, Schwann cells are present. Schwann cells are related to neuroglial cells. That part later we'll discuss. The types of neurons. If the neuron has one axon, If the neuron has one axon and many dendrites like this, it is called multipolar neuron. The multipolar neurons are present in the central nervous system, that is, brain and spinal nerve cord. In these bipolar neurons, the cyton has two processes. One is the axon and another is the dendrite. So, efferent process is the dendrite, efferent one is the axon. Such type of neurons are called bipolar neurons. These are present in retina of the eye. Next, unipolar neurons. Unipolar neuron has only one axon process, and this is found in embryos, not in adults. Then how it will receive the impulses? The cyton itself acts as the afferent structure and receives the inputs from outside. Next, pseudo-unipolar neuron. The pseudo-unipolar neuron, the cyton gives one protoplasmic projection. This projection divides into two. One is the short and another is the long. This is called dendritic branch. This is the axonic branch. Such type of neurons are present pseudo-unipolar neurons. These are present in dorsal root ganglion. And functionally, the neurons are divided into three types. Sensory neurons are efferent neurons, motor neurons are efferent neurons, and interneurons. What is that? Let us have a look. Assume that this is the central nervous system and this is the sense organ. From the sense organ, the dendrite is collecting the impulse and giving to the cyton. From this, one axon forms. So, this is afferent process or sensory process. The neurons which bring the impulse from organ to the CNS are called sensory neurons. So, these sensory neurons or the afferent neurons receive the impulses from receptors and supplies it to the central nervous system. You see this diagram. In this one, dendritic branches are very long, axonic branch is short. And next one, motor neuron, let us see. Motor neuron also called efferent neuron. This neuron has many dendrites. These are the dendrites. And from this one axon forms which goes to the motor organ. And such type of neurons are called motor neurons or efferent neurons which take the impulse away from the cyton or CNS. In a central nervous system, between the sensory neurons and motor neurons, there are some special neurons are there like this. These neurons are called interneurons. Interneurons link the sensory neuron and motor neuron. Now let us see how the impulse pass. This is the receptor organ and afferent process or afferent neuron and interneuron and motor neuron and motor organ or effector organ. This path of nerve impulse is called reflex arc. That is about the classification of neurons on the basis of their function. Functionally, these are sensory, motor, and interneurons. Structurally, these are multipolar, bipolar, unipolar, and pseudo-unipolar neurons. Next one, neuroglial cells. The neuroglial cells are Schwann cells. The Schwann cells are present around the axon. See, this is the neuron, this is the axon. These are the terminal branches. These are the dendrites. Around this axon, some cells are arranged. These cells are called Schwann cells. So, around one axon, many Schwann cells may present. These Schwann cells act like insulators. They do not allow the impulse or electrical signal to escape or to lose from this axon. Between these Schwann cells, here one node is there. This is called node of Ranvier. These Schwann cells, they form myelin sheath. Such type of nerve fibers are called myelinated nerve fiber. What is a nerve fiber? The axon surrounded by the Schwann cells is called a nerve fiber. If the nerve fiber has myelin, it is called myelinated nerve fiber. In a myelinated nerve fiber, myelin sheath is absent at the nodes of Ranvier. So, the Schwann cells, they form myelin sheath in peripheral nervous system. And similar to Schwann cell, in central nervous system, oligodendroglia are present. These oligodendroglia form myelin sheath in central nervous system. The peripheral nervous system, Schwann cells are forming, and in the central nervous system, oligodendroglia or dendrocytes will form the myelin sheath. Next one, astrocytes. These are the astrocytes. Two types of astrocytes are shown: protoplasmic astrocytes, fibroastrocytes, whatever they may be. These are acting a barrier between the blood and the brain. Assume that this is the brain and this is the blood. So between the blood and the brain, they are forming one wall. So infections cannot reach the brain. That is the role of the astrocytes. They form blood-brain barrier. And next one, some ependymal cells are there. In earlier classes, we discussed that the brain is not solid. It has so many cavities. Those are called brain ventricles. This brain ventricle is lined by some columnar cells. These are called ependymal cells. These columnar cells have the cilia. And these ciliated columnar cells are the ependymal cells. These are useful for the circulation of cerebrospinal fluid. Microglial cells. These microglial cells are mesodermal. Rest of them are ectodermal. These microglial cells are phagocytic cells. These are similar to the monocytes of the blood or the osteoclast cells of the bone. And many there, like in the liver, Kupffer cells are there. These all belongs to one category. Now, let us try to understand what is a nerve. This is a neuron. It is the axon. The axon with Schwann cells is called nerve fiber. This nerve fiber is covered by one endoneurium. Such type of nerve fibers are arranged in a form of bundle that is called funiculus or fascicle. And this fascicle is covered by one more dense irregular connective tissue sheath. This is called perineurium. The bundle of fascicles are wrapped by one more layer, connective tissue layer called epineurium. So epineurium surrounds the funiculi or fascicles. So the bundle of funiculi or fascicles is called nerve. The nerves also different types: sensory nerves and motor nerves, mixed nerves. The sensory nerves take the information from organ to the brain, and motor nerves conduct the impulse from brain to organ. Mixed nerves do the same function, both the functions. Thank you for visiting the Chocolates class.